Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of Kiss Organics. This is the podcast where we discuss the cutting edge of growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. Before starting the podcast, I wanted to share about the Cannabis Cultivation and Science Conference that we are hosting in conjunction with Collective Elevation in Bozeman, Montana. The event is happening on September 5th through 7th, 2025, and we are kicking it off with workshops from the Bug Lady, Suzanne, and Dr. Greenhouse, followed by a facility tour, and then a bunch of excellent speakers that were former podcast guests such as Jeremy Plum, Dr. Michael Brownbridge, Les Evans from DRAM, and many more. It's going to be 3 days of talks and networking. Then on the 7th, the Montana Living Soil Alliance is hosting a tasting event that will be included in the price of the conference ticket. The tasting event will be a private event with cannabis donated by the Living Soil Alliance as a way to have an amazing sensory experience and see what premium quality Montana Living Soil has to offer. Early bird tickets are $200 and limited to 200 spots, so be sure to grab a ticket if you're interested. Tickets are now on sale at kisorganics.com. Bozeman in September is pretty beautiful and is a popular spot, so best to lock down your ticket and accommodations early to guarantee a spot. My guest this week is Michael Gutensohn. Michael is an associate professor of horticulture at West Virginia University. He earned his diploma in biology from the Julius Maximilians University in Germany, where he specialized in plant physiology. While pursuing his PhD at the University of Cologne in Germany, and during a subsequent postdoctoral position at the Martin Luther University in Germany, Dr. Gutensohn worked on multiple projects studying metabolite and protein transport processes in plants using biochemical and genetic approaches. While at Purdue University for a second postdoctoral position, he was introduced to his current research topic, the biosynthesis of volatile organic compounds, VOCs, in plants and their various biological functions. His research interests include plant-insect interactions, such as plant defenses against herbivores and pollinator attraction to flowers, flavor compounds in horticultural fruit crops, specialized metabolites in medicinal plants, classical genetic and metabolic engineering approaches to introduce or improve desired VOC traits into crop plants, and mechanisms regulating the flux in complex metabolic networks involved in terpene formation in plants. Dr. Gutensohn is also involved in steering the operations of the Analytical Biochemistry Core Lab in the Division of Plant and Soil Sciences. Now on to the show. Well, thanks for coming on the show today.
Yeah, thanks for having me. I'm excited.
Yes, so I saw an article about your research and reached out to you, and you were kind enough to get back to me, and we've talked a little bit offline. But I was hoping you could share some of your research and findings with our audience and what you've been working on at West Virginia University.
Yeah, sure. Happy to do so. Well, basic approach of what we're doing is that we take a little bit of a step back from looking at hemp and cannabis more from the human perspective. And maybe look a little bit more at it from the plant side of things. And we're trying to understand why certain things as terpenes and cannabinoids are being produced in hemp. The discipline that I and my lab are working in is also very often called chemical ecology. And so it takes exactly this approach. Like, we're trying to understand why plants produce what on the scientific side of things is called secondary or specialized metabolites. And in general,
you define that for listeners as to what falls under secondary metabolites within the constituents of a plant?
Yeah, I know this is probably a term that most people are not so familiar. So in general, we talk about either primary metabolites or secondary/specialized metabolites. Primary metabolites is something that most people are pretty familiar with. This is your carbohydrates, your carbs, how people call them, your lipids, and your proteins. Yeah. So this is what we need in our nutrition, basically, and you should have a little bit of a balance there. This is what's primary metabolites, and they're produced by all organisms, including plants. What's specific about plants is that they also produce a large set of other metabolites in very small quantities very often, and those are called secondary or specialized metabolites. And that has a little bit to do with the lifestyle of plants. If I scare you or one of your listeners, they probably would run off and flee. Yeah. Same thing for an animal. Yeah. If an animal is in a stress situation, one of the easiest ways to get out of it is just to run away and flee. A plant is rooted. It can't run away. And so it has to fight back basically at the spot where it is. And that's why plants developed into being absolute masters in producing a lot of very interesting and could also say nasty chemicals. Yeah. So a lot of these specialized metabolites plant produce because they have to somehow deal with their environment and to a large extent with their what we call a biotic environment. So basically with other organisms, with other plants, with other microbes, and of course also with animals. And among animals, I think for plants, the most important ones are the insects because that's number-wise probably what they face most. And so for doing that, they produce these metabolites, and that's no different for hemp. So what we humans use or are interested in hemp are actually chemicals that are there for a purpose to help the plant, not necessarily us. And that's kind of the little bit the philosophy or approach that we're taking. We're trying to kind of ignore to some extent why humans are interested in hemp and cannabis and rather try to understand why is the plant producing these things in the first place. Yeah. Because it actually helps better understand how these things are regulated and produced in the plant, when, where, and how. And then that creates a lot of very interesting and useful basic knowledge that then you can apply later on again. So this is a little bit in a nutshell our approach there.
So you studied factors that increased secondary metabolites in hemp, specifically, the most common ones that I think of when I think of that would be CBD, THC, and terpenes would all fall as secondary metabolites. Are there any others that you were looking at in some of your research?
Yeah, so I mean, when we do analysis, we generally look at cannabinoids, but not just the ones that you mentioned, although those are, of course, are the most abundant ones. We simply look from the point of view of how much amount-wise is there. And then the other big group of compounds that we're looking at are the terpenes, so the essential oils, basically. What you normally perceive as the scent or the smell, because they go into the gas phase and you can sniff them. So that's the two major classes. But we in our analytics, we inevitably don't just see a few compounds there, like you mentioned, THC, CBD, and some of the other well-known cannabinoid compounds. But when we do an analysis, we currently are in a situation where we can roughly see about 50 different cannabinoids. Although, I mean, if you look in literature, there's over 100 cannabinoids known by now. It's technically quite challenging to detect all of them. I mean, we over the last couple of years, we've definitely improved in our analytics and can detect more things now. And the same applies for the terpenes. I mean, we see basically everything that's there. The question is just, can we really identify undoubtedly everything? And I'm always a little bit on the cautionary side there. I don't want to lean myself too much out of the window and claim I can identify everything. But we see a lot of things there. But that's the two major groups of compounds we're looking at. And yes, you're right. I mean, what you said before, that we're particularly interested what induces the formation of these things. And that is actually was sort of the onset of a USDA grant that we were awarded in 2020 as one of the first ones that got actually federal funding for working on this. And the major question that we were trying to address in this project was the so-called going hot problem. So environmental factors triggering an increase in the THC content that would push industrial hemp from its legal status to surpassing the 0.3% THC content, literally turning it from a crop into a drug. And we wanted to understand or identify some environmental factors that would do so, and then also start understanding a little bit the metabolic and genetic regulation that's behind that. So why is that happening?
Now, you were working on hemp. A lot of people listening to this podcast are growing cannabis. Um, my question to you would be: is it fair to assume, with the similarities between the crops, that there may be some overlap in terms of your results having similar effects on cannabis plants as you're seeing with hemp?
I got to be a little bit careful there. So I can definitely say we also tested seed hemp varieties, and not just CBD or CBG varieties. And those, so seed hemp and CBD varieties behaved in the same way. They both saw an upregulation of THC production under certain stress conditions. Of course, at completely different levels. Yeah, I mean the THC level in a seed hemp variety is fairly minimal compared to what you find in a CBD variety. But in principle, in both cases, but with both different types of hemp, we saw an increase in THC. You're not going to run anywhere close to being in sort of risking or surpassing the legal THC limit with a seed hemp variety because it's so little. But
Mhm.
in principle, the induction you could see in well as well there. So based on that, I would probably conclude that yes, there's a similar regulatory mechanism in play, just that you're starting from a different baseline, so to say, and of course that determines how much you get. I can't say if that's the same thing would happen in cannabis. Um, as you probably know, the problem is that we can still not work with cannabis because it's on the
Sure.
research side in academia because it's still illegal at the federal level. And since we do get federal funding, we have to follow federal rules and laws and regulations. So working with cannabis that has a higher THC level than 0.3% is off limits for us and will stay off limits as long as there's no change in the legislation on the federal side of things. Um, so that might be confusing for some people if they think, well, but wait a moment, I'm in my state where I'm operating, I'm a grower. It's legal to grow it here and sell things. But the point is we get federal funding and that determines what we can do. So it's basically depending on our funding status. So universities cannot work with cannabis because of the funding situation. That limits us. And so I can't tell you anything about cannabis because I'm simply not in a situation that I can work with.
I understand. So what you're saying is it's likely, your hypothesis is that it's likely but not definitive that you would receive similar effects based on the similarities of the two plants.
I mean, I would, based on the results that we have, I would, if I would ever come in a situation that I could work on cannabis, my hypothesis would be that I would expect
Sure.
the same regulatory mechanisms at play. Um, but of course, that would have to be tested. The question, of course, is, I mean, what I could envision, or what you could put up as a counter argument there is like, well, in cannabis, you have already such a much higher THC level. Maybe you're sort of maxing out at some point, yeah, and
Hmm.
then you can't increase it even more because there is already so much that there's other limitations that basically would limit it from being further increased. Yeah. Um,
Yeah.
so that could be one thing, but I mean it's pure speculation at this point. Before we're not in a situation to test that, you actually won't know. Yeah.
I I want to talk to you about plant stress, but before we do, because you brought this up, there was some, I think it was Dr. Daniela Vergara at Cornell, I believe, that I talked to, that mentioned this idea of a dilution effect, where a plant has the genetic capability to only produce so much, so many cannabinoids or so much THC, and then once once it reaches a certain level of biomass, that THC level in theory would be diluted across more, greater surface area because it's producing more biomass. So in theory, as your yields increase, some of the concentrations of some of these secondary metabolites would in theory go down.
Yeah. That's one thing you could envision. Of course, I mean, the question is like, how do you achieve your final THC yield, so to say, if you want to call it that way? You brought up biomass. That's, of course, one point. I mean, the more biomass or flowers a plant produces, of course, the more yield you will have there. And of course, that can be influenced badly by the growth habit of the plant. So that's one factor. I would bring up one other factor or argument in there, and that is, I mean, I'm from my background, I'm a plant biochemist, and so of course, I always look like more at from that perspective at it. And there are other factors that can limit production. And you have to think about any of these compounds. They're produced from certain precursors that need to be provided from the rest of the plant. And at some point, you might just reach a point where there's limitations on that end. The plant can't just provide so much building blocks, so to say, for making cannabinoids as it can without at some point sacrificing other things. Yeah. And then it's a trade-off between producing more, in this case, would be cannabinoids versus kind of needing other things as well. And so it's very often this kind of like what gives the plant more of a benefit, producing more cannabinoids maybe versus producing something else in a larger quantity. So we we very often talk when we in the science side of things, when we talk about metabolism, we very often talk about networks rather than sort of linear pathways. Yeah. Um, and that's anyway where science has moved in the last 10, 20 years on the biochemistry side, that we understand that everything's kind of like a huge network connected with each other. And although very often we just look at one final product because that's what in a particular project you're interested in, but you always have to keep in mind that is not isolated from all the rest that's going on there. And so you have to kind of consider that as well, that you're rather looking at one tiny little aspect of a gigantic network of things. And just because if you're regulating one things, there's 250 other things happening in parallel, and you can't ignore
Yeah.
that. Yeah. So even though you're kind of interested in bringing up the level of that one thing that you're interested in, you have to consider that that's happening at the backdrop of everything else that's happening there as well. And so there might be limitations that come rather from this whole network that say like, well, that's enough now. We need to do other things as well here, because otherwise the plant's not doing well either. Um, so
So
that could be one other factor sort of limiting things at some point.
yeah, so that has me thinking. So what you're saying essentially is a plant has a certain amount of resources, limited amount of resources, and it allocates those resources based on its goals, either plant defense to protect itself. In the case, like you mentioned, if someone scared you, you could run away. Well, the plant would have to produce these secondary metabolites to protect itself from insects, environmental conditions, whatever, or as a way of a continuation of its species by reproduction, essentially. Correct. So if I'm understanding correctly, we have a limited amount of resources, and that plant is going to allocate those resources towards one of those goals, whether it's reproduction, protection, growth, overall growth. And so it's a constant, I don't know what the word I'm looking for here is. It's just a constant process, I guess, within plants.
Balancing these various different things out. Yeah, it's constantly a balance, and if you chip, if you raise one thing up, you inevitably going to lose at some other end. Yeah, there's only so much in total available, and ultimately, if you want to really go to all the way down, I mean, like, um, ultimately, anything that a plant produces and does comes from photosynthesis. And the photosynthetic capacity of a plant basically limits how much is there, how much resources are there. You can't do more than what it fixes in photosynthesis in form of fixing carbon dioxide and producing sugar molecules initially, or how much nitrogen you can take up from the soil or other nutrients that you need. That ultimately limits things. How much is there, and that limits what you can make out of that. Yeah. So there's an input and there's an output, so to say, and that ultimately needs to be in balance. And all these things limit in the end how much you can produce of a certain compound, basically. And yes, you're absolutely right. I mean, growth, reproduction in form of seed production, or in other crops, it would be fruit production. Um, that's other factors that play into that. Yeah. And you can't just ignore that. It's just there. Yeah.
So one of the examples I think of that kind of highlights this is the idea of that plant that you threw out because it was sickly, or a seed that got lost and ended up in your compost pile, and it was neglected all season long, and yet this plant was able to survive and not thrive because it probably had nutrient stress and drought stress and maybe heat stress, but it was able to survive through all of these things. So it was very resilient. At the end, when that plant is ending its life cycle, what I've seen is higher levels of secondary metabolites, higher THC, maybe higher levels of terpenes, all of these secondary metabolites, but the yield is dramatically lower. It's a sickly plant. It's not necessarily healthy, but due to all of these stresses from being neglected, um, we tend to see higher, people will say the best cannabis I ever smoked or grew was this poor plant that was, horribly stressed. What are your thoughts on that? Is that sort of fit with this idea of allocation of resources, as the plant was just trying to survive?
Yeah, and it's not necessarily just the plants survive, it's the species survive. Plants don't function necessarily at the level of individual plants. They try to survive as a species. And so what plants very often do, if they're in what you described like a nutrient deficiency, or so, like just not a very good environmental condition, which yes, you can create in your greenhouse, in your growth facility, yeah, because you neglected it or overwatered it or whatever, something went wrong. Um, One of the important things that plants very often do is they try to produce seeds in the hopes that the next generation actually finds better conditions. Yeah.
Mhm.
And so it will try to somehow get through and produce, even if it's just a few flowers and a few seeds, but it basically, in the hopes of maintaining itself as a species, it produces seeds, which is the next generation, and then hopes that those seeds are spread somewhere in a place where there's better conditions, or next season is the weather is better, yeah. There's more
Mhm.
rain or better conditions, or the seed is carried to some other environment where the soil is more fertile or something like that. So yes, you always have to look at that. Plants function in a different way, and that's something goes back to what I said at the beginning. This is you if you want to understand what plants do, and if it is a crop plant, and hemp or cannabis are crop plants, applies to a crop plant in the same way like to any wild plant. Yeah. Sure, crop plants, there's a few things that are different because we humans over hundreds, if better say thousands of years, have sort of selected certain things, and certain traits have been manipulated in certain ways. But a lot of this basic regulation is still there. It's not gone. Um, and if we want to understand our plants, then you really have to put yourselves in the literally have to put yourselves in the shoes of a plant to understand how it's actually functioning. And that goes back to what I said. Sometimes you have to, I think, for understanding hemp and cannabis, I think you sometimes have to take a step back and say, like, well, I know our final goal is to grow hemp or cannabis for the purpose of getting cannabinoids or other purposes, but why are things happening there in the first place? And for understanding that, you have to take the human component out a little bit and look rather from the plant side of things. And that's what we're trying in my lab and in our research. We're really trying to go back, and it's like, okay, why are these things happening in the first place? And what are the underlying mechanisms that are there? Yeah. And then we can kind of understand. One factor with any cropland is that you have to consider, well, there was human domestication and then cultivation and breeding happening for eventually thousands of years. So of course, that's not a wild plant anymore. So some things were selected or changed, but a lot of the basic regulation is still there. That hasn't gone away and will not go away. Yeah, it's still there. Yeah. So
So yeah, so you studied essentially what would cause a plant to go pot to create more of these secondary metabolites. Um, Before we go into what your findings were, can I have a question about plant stress? So we've been talking this whole time essentially about plant stress. What stresses plants out? And it's a variety of things. Off the top of my head, you can have heat stress, you can have cold stress, you can have a variety of different types of environmental stress like drought stress, overwatering, and then you have your, biotic stress like insect pressures, things like that. Herbivory with an insect eating the leaf itself. Are all stresses to plants the same, or do they have different effects on the plant?
no, they're not all the same. That for sure. So in I mean, plants need to respond to all stresses, and we basically you outlined it perfectly there. So in principle, you can categorize things in two major buckets. The one is abiotic, so anything that is in the environment that's not another organism, and you mentioned water stress, heat, cold. You can, I mean, even light stress in the sense of UV or too much light. So there's all sorts of factors that can involve that. And then, of course, nutrition, lack of it, or too much salinity, soil conditions. I mean, there's a lot of things that can go into that. Acidity or sort of pH value of water. There's a lot of things there. So that's the one side of things. And then the other side, yes, as you mentioned, is the biotic side of things. And that's basically microbes on the one hand side, which very often are like either pathogens that harm the plant or they're helpful if they're like fixing nitrogen or mycorrhiza that help taking up nutrients. And then, of course, it's animals. And again, they can be beneficial or they can be sort of harmful. Herbivores, you mentioned, yes. But there's also beneficial insects. I mean, growers will know that your beneficial insects in your greenhouse and your growth facility to kind of fight pests. And plants actually very often actively attract these beneficial insects for helping them. But how a plant responds to these different abiotic and biotic stresses can be quite different. I mean, you can't generalize. Every individual factor can have a different consequence. Yeah. So plants, some of these specialized or secondary metabolites help the plant actually sort of cope with some of these abiotic factors. So you have certain things that almost work like a sunscreen. Yeah. So they protect plants from too much light or light stress. Yeah. Other things very often sort of help with sort of some of the consequences of some of the stresses. Yeah. Um, and so cannabinoids are maybe not necessarily falling directly in that category, but of course you're right. You mentioned that before that sometimes plants, hemp plants, cannabis plants respond with sort of an alteration in their cannabinoid level if they experience some of this abiotic stress. But that sometimes could be rather kind of an indirect effect. If you're changing sort of a supply of nutrition, if there's too much or too little, then that's kind of like if you see a change on the cannabinoid level, that might be rather kind of an indirect effect that overall metabolism of the plant is affected, and then that's affecting cannabinoids as well. So that's the one thing that can happen there. With the biotic stress, that's a completely different thing because a lot of these secondary metabolites are specifically produced and very often upregulated to deal with these stress situations. So with biotic stress, with being attacked by pathogens or pests, that's their one of their main purposes. Yeah, and that's no different for terpenes or cannabinoids. That's why they're there.
So, So basically what you're saying is all stresses are not the same and don't have the same plant effect. And what we're getting as in terms of cannabinoid effect is actually not the primary mechanism or necessarily what the plant is focused on when it's creating these. Is that am I understanding kind
On
of what you're
on
saying?
the Well, I mean, we haven't done much on the abiotic side, but of course, I'm aware that there's quite a bit of literature out there. But I would have a little bit of a hard time seeing how a cannabinoid molecule could help a plant um, with dealing with some of these abiotic stresses, sort of directly dealing with it. Yeah. There's other compounds where you can make a direct connection. Yeah. They produce some of these things. Like I said before, it works like a if you putting on sun lotion or sunscreen, basically. Yeah. Cannabinoids don't do that. Yeah. Um, First of all, from where they're being produced. Um, and second of all, also from their chemistry, I would have a little bit of a hard time seeing how that would
So
work.
so you're saying like drought stress, like increasing terpene levels in a plant or increasing CBD does not directly help a plant that's experiencing drought stress. So it may be some sort of secondary mechanism that occurs
It could
as
be
a
either a
result.
secondary mechanism, sort of kind of like just something in metabolism is altering, and that kind of results in some consequence in cannabinoid or terpene formation. The only thing you could argue for is a plant is in generally stressed by an abiotic factor. Of course, it's potentially also at risk of being more susceptible to biotic stress. Yeah.
Yes.
So you're anyhow stressed, you're weakened because of some environmental factor. There's a drought or there's something else going on in your environment. So you're basically in a weak, um, susceptible status. And that makes you more prone for being attacked by pests or pathogens, basically. And then in that context, of course, it would make sense to upregulate some of these compounds because basically you're still trying to defend yourself. Yeah. But again, it's not the abiotic factor that you directly are kind of like dealing with. It's a consequence of the abiotic factor weakening you, so to say, as a plant. And then that results in potentially you being more at risk of being prone to other stresses. Yeah.
Yeah. So you're saying as the plant receives drought stress, it thinks, "Oh my gosh, I'm weaker. I'm more open to pests. So I by increasing my terpenes or my cannabinoids, I may better protect myself from these pests that are more likely to hit me now that I'm more susceptible." So it's
Yeah. I mean, that's the biological explanation. That would be the biological benefit of doing that. Yeah. Sort of
yeah.
you're increasing your likelihood of surviving or reproducing at least to the extent that guarantees the survival of you as a species, basically. Yeah. So there's you always have to look at the biological purpose of something. And so yes, you can draw that conclusion. I'm not saying that that's what it is because I don't know, we haven't looked at that. But in other plants, that's clearly there is a connection between abiotic stress and some of these responses, which is in some cases it's not clear that the the increased production of some of these secondary metabolites would help dealing with this abiotic stress, but it basically protects the plant from potential other things in that situation.
Now, a popular term or concept in the last few years has been this idea of crop steering, which is, I don't know if you're familiar with it or not, but it's essentially intentional stress that you're giving to the plant to create a certain response. So, some examples of this, because cannabis growers have tried everything over the years, would be what they call drybacks, essentially intentionally withholding water to create drought stress at certain stages of plant growth, or withholding nutrients, or getting closer to harvest. I've heard of people um, reducing CO2 and reducing light levels to lower stress, but then on the flip side, cooling the rooms that they're in as a way of introducing cold stress. Do we know what sorts of targeted stress levels will have, what mechanisms create this effect?
That's a complicated question.
It is.
Um, no, I mean, in principle, I can see why people would try these things because, of course, whenever you're changing an environmental factor, and that's essentially what you're doing with all those things that you were mentioning there. Of course, the plant will respond to that in one or the other way. So yes, I mean one of the first things that I would bring up in that point, for example, and I don't know if there's sort of more trustworthy documentation or scientific work on that for hemp. But of course, you have to think about some of this might also regulate flower induction, how many flowers you get, the ratio between flowers and vegetative growth. It's kind of like you're manipulating the growth of a plant in the first place, yeah. So that logically has some consequences there. So that's one thing. And yes, I mean, like I said before, I mean, certain abiotic factors will have consequences on the level of metabolites as well. So yes, and I mean, I'm very well aware that people probably have done a lot of stuff just by trial and error and trying to play around with things, with conditions and see what the consequence of that is. Yes. And that's not much different than what we do on the science side of things. I wouldn't say we play around. We normally start off with a clearly defined hypothesis, but in the end, we don't know things, and then we test things. Just that we do it under much more, let's say, a little bit more controlled conditions, and we do it not just one time, but we do a lot of replications and very thorough data collection, and then data analysis later on to really kind of verify or reject a hypothesis that you might have had before there. But yes, I'm not
So,
saying that that's not reasonable to assume that some of these things would lead to respective consequences in the plant. Like I said before, I mean, we were kind of considering abiotic factors in our work a little bit, but since the initial results we got in the first year or year and a half were much more promising and interesting on the biotic side of things, we very quickly then decided to primarily focus on that, in particular because that is also a little bit more in alignment with other projects not related to hemp and cannabis that are ongoing in my lab here. In generally, I work a lot with plant-insect interactions, and so logically, once we saw respective responses in hemp plants to insects, we kind of focused primarily on that.
I want to talk about that next. But one question I want to just propose to you is something that the sort of my approach to understanding this is plants all have this genetic potential. And when we're cultivating, we're trying to get them to reach this potential. And then any sort of stress that they have, while it may increase secondary metabolites, it could also impact yields negatively because the plant has a certain amount of resources it can allocate. Now, I'm under the philosophy, and please feel free to correct me on this, that every facility, grower, cultivation I've ever been to has plant stress just naturally occurring. We haven't perfected this in real. I mean, in a growth chamber, maybe you're able to get a lot closer to this, but in the reality of cultivating it commercially or even at home, we're naturally going to have some of these stress events occur already. And so my goal as a cultivator is to create the best environment I can for the plant to reach its genetic potential and assume that it's still going to have good secondary metabolite production. Um, do you have any thoughts on that approach versus this idea of, more targeted stress events?
Um, I think in general, the take of providing the best environmental conditions, if you want to say so, is for sure the best way. You don't want to intentionally harm your plants because, yes, you're right. I mean, if you do that in the wrong time or in the wrong way, you simply will sacrifice your yield overall. Um, some targeted sort of treatment, if you want to call it that way, at a certain time point to sort of increase the level of certain things that you want to sort of achieve there. Yes, that makes sense. But sort of from the get-go, intentionally stressing your plans all the time, I would really not think that that would be a particularly good idea. Yeah. I mean, initially, you want to make sure that your plant grows up properly, is in a good, healthy condition, good growth stage. If you then at some point set sort of the right trigger through some treatment of your growth conditions, your environmental conditions, yes, you might achieve something there. But I mean, this is similar things you do with other crops as well. Yeah. I mean, but you got to be careful with that. I mean, like just sort of trying to harm your plant intentionally for an extended period of time is probably not a good idea there in the first place. Yeah.
So what you're saying is we may find that and this is another thing, is we have such genetic diversity in cannabis that we can't make the assumption that all plants are going to respond the same because of the diversity that we see. But we may find that 2 weeks out from harvest, for example, extreme drought stress or extreme cold stress may have a positive effect on secondary metabolite production without having as large of an impact on yield. And that might become best practice down the road, but we're not there yet.
Yeah. Yeah. I mean, keep in mind, you keep your plants in a vegetative state for quite a while before then they start flowering. There's no
Mhm.
sense in stressing your plants while they're in the vegetative state. I mean, you want to
Good
have a
point.
good baseline, so to say, of vegetative growth because that's the resources that you ultimately then can remobilize and put into flowers in the end. Yeah. If you're having a weak plant before, well, what's the sense of it? Yeah. There, that's not going to work out well.
That's a really good point. Yeah. So we definitely don't want to create any stress early on in the plant's life. Okay, well let's talk a little bit about your research. So we kind of hinted at it. Tell me a little bit about what your sort of methodology was and what your results were.
Yeah, so I mean, this goes way back to what I said at the beginning, a little bit this chemical ecology approach. And of course, one of the major stresses that any plant will face in its lifetime is basically biotic stress, um, and in particular, insects, herbivory, basically. Of course, we can talk about herbivory in a little bit of broader sense. I mean, not herbivory is not just insects. And I think this is something we might want to consider a little bit more when we talk about hemp. We can get to back to that in a few minutes. So herbivory could be also by other animals, by mammals, by little rodents or rabbits or goats or sheep or something like that. I mean, if you consider the size of a hemp plant, it's not a tiny little plant. It's actually a quite substantial plant. So you can imagine that other animals would feed on a hemp plant as well. Not sure if it the flowers, but definitely somewhere on the plant. Yeah. But definitely the other major factor that kind of harms plants in that respect are all sorts of insects. Yeah. And so and that's normally why plants produce a lot of these specialized metabolites, basically fending off this herbivory. So any herbivore that consumes some of the leaf material, the plant, the vegetative part, even if it's in the roots, um, would be facing or would be exposed to such specialized or secondary metabolites. And that's no different for a hemp or cannabis plant. Yeah. And so one of the early on experiments that we did was we kind of initially mimicked herbivory, and you can do that by some chemical treatment basically that is a signaling compound that's actually a plant normally produces itself when it's exposed to herbivory. And then we measured terpene and cannabinoid contents, and we saw already that that led to an upregulation. Yeah. And so that was a first clear indication, oh, yeah, they're responding to that, which was not surprising. It's just easier to do that because it's basically you spray a chemical on the plant and you can see the response, and you don't actually need the actual insect feeding on the plant. But logically, that was our next step. Once we saw these first preliminary data and seeing that the plant was responding to that, then of course we looked for a an insect system that we could use on hemp, and we kind of looked around and talked with some people from various extension services to see like what are real insect pests that you're seeing in hemp and cannabis cultivation. And then, of course, we also looked for something that's actually relevant and easy to get your hands on in our region here in Appalachia. Um, and it turns out actually that beet armyworm was a real pest that was an issue that growers had to face and deal with. And at the same time, there's a small company that rears these insects, these larvae, and we can simply buy them from them and get them shipped here. And there's no legal restrictions. Sometimes with pest insects, you got to be a little bit careful. You can't just move them over state lines. But in case of beet armyworm, that wasn't a problem because it is already a natural pest here. So we're not introducing something that wouldn't be here. And so we finally settled on using them. So we get little caterpillars that are pre-reared already. We keep them here for a little while, and then we actually allow them to feed on our hemp plants. And they actually do quite a bit of damage in a relatively short amount of time. They start off feeding on leaves, but then at some point they also move to the flowers. We have damage on leaves and we also have damage on flowers. And then under very controlled conditions, how kind of determined how long we had to leave them on there, and then measured cannabinoid contents and indeed saw that cannabinoid production was quite significantly upregulated. And actually upregulated to a level where we would be in violation or could get into violation if we would be growers of the legal limit. Yeah. So this clearly indicated that insect herbivory, if you're growing CBD hemp, can be a problem for you as a grower. So I mean, one of the basically easy take home messages from that work was already just yes, you got to scout for insects, pest insects if you're growing, and you got to take care of that, which I know is not easy because depending on what time life status or of your plants, you probably don't want to spray much because I mean then you're running into problems there again. But scouting early on, making sure you don't have a lot of herbivory, and if so, take the right measures is definitely one of the core things. So this was the first finding, but then the story kind of developed a little bit more. Um, we saw that compounds, major cannabinoids like THC, CBD, CBG, some of those things were upregulated. But then we saw, since we're in our cannabinoid analysis, we're not just seeing those major cannabinoids, but we're seeing a lot of other so-called minor cannabinoids as well. And what we were surprised is to see that those were also upregulated and actually also not all of them, but some of them, and some of them actually quite significantly. So things that were in our analytics before tiny little peaks and difficult to detect were very detectable afterwards. So significantly upregulated and increased. And that, of course, we got really excited about that and sort of like draw our attention to because of like we were wondering like what the heck is going on there. And terpenes were also upregulated, but not all of them, and also not to an extent that was really very extreme. Yeah, it was a little bit of an upregulation, but nothing really that was particularly didn't knock me out of my shoes, put it that way. And that was a little bit surprising because we originally had my expectation when we started this was that rather the terpenes might be affecting insects because that's what they do in a lot of other plants. So I have a
Yeah.
couple of other projects where we do similar things, and very often terpenes or some terpenes are the ones that are actually really kind of affecting insects. And so my initial assumption or expectation or hypothesis was that, yeah, in hemp, it's probably also going to be terpenes being involved in that. And from the first results, it did not look quite like that. I don't want to exclude that the terpenes play no role, but it was that the response on the cannabinoid side was much more significant than what we saw on the terpene side, and that was a little unexpected.
Okay, I have a few a few questions from that. Um, First question is in relation to the terpenes, are we talking, a statistically significant increase?
Yes, yes, for some of them, not all of them, yeah, but for some of them it was a significant increase, but it was from the extent was not as dramatic as some of the responses we saw on the cannabinoid side. In case of some of the cannabinoids, we see like a fivefold increase
That was my next question. So you're
with
you're
with
saying
with
massive increases. Okay.
really massive increases. Yeah.
Wow.
I'm not saying THC necessarily or CBD, but some of the other things we really see like quite significant increases. And with the terpenes, we maybe see a 1.5-fold increase or something like that. Yeah. So by far not as dramatic. Yeah. I'm not saying that they don't play any role, but it was really not kind of quite what we had expected initially. Um, so that got us really kind of like a little bit wondering what the heck is going on there. Um, and of course, whenever you find something like that, of course, we want to understand why that biologically makes sense. Why do you have these upregulation of cannabinoids there? What is it doing? And that brings me back to what we were talking about before with the herbivores. Yeah. So if you have an upregulation of THC or CBD and your herbivore is a mammal, I'm not saying a human, but a mammal, a little rodent, you can explain how that might affect that herbivore. Yeah. They do have the same cannabinoid receptors that we have, and I'm not saying they're getting a high, but they're for sure going to have some experience there or some exposures to it, and would probably kind of reconsider feeding on that plant the next time, potentially. So you can see how that would work. Interesting thing is with insects, they don't have the cannabinoid receptors that we and other mammals have. And that, of course, brings up the question like, then how does it affect insects? Are there cannabinoids that affect insects?
Is so one thing I was wondering is how target is are plants able to distinguish in regards to like how targeted is their approach, defensive approach? So like for example, can a plant distinguish between thrips damage and caterpillar damage? And maybe for thrips, it may increase terpenes, whereas caterpillars, it focuses on cannabinoids. Is that a possibility, or are plants not able to tell if I'm cutting it with scissors or if it's, I mean, I assume there's some sort of chemical interaction happening, like you mentioned, with being able to spray something to get a similar plant response.
Well,
How
the
does that
first thing
factor?
the first thing you can distinguish on the insect side is what type of insect you have there. And you brought two examples up there. The one is caterpillars, which fall into the group of so-called biting, chewing insects. So they actually really consume leaf tissue. They bite into tissue and munch it up, basically. Um, The other example that you had was thrips, or you could add a couple of these other things like aphids,
Mhm.
white flies, spider mites. Those are generally kind of considered as piercing, sucking insects. So they either feed on the vascular tissue or the content of it, or they actually suck out cell content. So they puncture individual cells and really suck the content of the cell up. That is a completely different type of feeding, so to say, and the damage that you're seeing is a completely different one. So we did not see much response with piercing sucking insects, although we have not really used them a lot. I know some people have seen effects when they were using aphids. So I don't want to exclude that, but we haven't seen that in our analysis. So we primarily kind of concentrated on biting, chewing insects that really have consumed tissue. We actually have used a couple different ones in the meanwhile, and we see the same response. So we're pretty sure that that is pretty uniform there.
Hmm.
Um, and that is something that the plant can distinct. Yeah, I mean there is a difference in the response of plants if they're sort of experiencing herbivory by biting, chewing insects versus a piercing, sucking. Even the sort of the signaling within the plant is different when they're exposed to these different types of insects. But you're right. I mean, what the other thing that you just mentioned before is so what the plant can perceive is not just when you have a caterpillar or a beetle nibbling around there, it's not just the wounding of tissue. The plant can actually recognize the insect side of things as well. So plants do have, per se, in general, have receptors for the saliva of insects. So it's not
Wow.
just the wounding. Um, there is publications not from hemp, but from other plants where plants can perceive the feces of insects. So very often, if there's a caterpillar or a beetle or something like that roaming around on a plant, they leave their they basically defecate on the land. They leave their feces behind, and there's things in there that the plant can recognize. So plants can recognize quite a lot of things, um, sort of traces that an insect would leave behind if it's on a plant and feeding there.
So someone might develop a caterpillar feces spray someday that we could spray on our plant to get upregulation of secondary metabolites. In theory, I
In
mean,
theory, yes. I can tell you from research that people have actually collected the saliva of insects and have diluted that and sprayed it on plants, and that led to a response with even without the wounding, but you can combine it with the wounding and then you even get a stronger response. So there's
wow.
some good literature out there, not from hemp necessarily or cannabis, but from other plants. But I mean, this is by now it's pretty accepted that that's a pretty general response of plants that they're capable of recognizing these sorts of things. Yes.
So one other thing you touched on, I just want to highlight and make sure I understand is this idea that chewing insects, like the caterpillars that you worked with, create a much higher response in terms of secondary metabolite production than sucking, piercing insects. So the practical application of this might be if you are seeing aphids in your hemp grow, that may be less concerning than caterpillars or rodent damage or other things in terms of the THC response that you may see from that herbivory. In theory, just pure hypothesis.
In theory, I mean, like I said, we did not see a response when we mimicked aphid feeding, so to say. And again, we never used aphids. We mimicked this kind of feeding with some treatments there. We did not see a response there, but I'm aware that there are some reports from other groups where they did see a response of plants upon aphid feeding. So I don't want to completely exclude that. I can't just say we did not see it, but that doesn't mean this could be again your what you mentioned before, your genetic diversity basically. Maybe there are some varieties where you see that response and others not. Of course, we only used a few hemp varieties and not too many. I mean, it needs to be manageable for on our side. Um, I mean, I wouldn't generalize it as you said. I mean, you might be right that I would be more concerned about sort of herbivory by caterpillars or beetles or some biting, chewing insects. If you're concerned about your THC level, I would not say that I would not be worried about an aphid infestation.
Sure.
The problem in general is that piercing, sucking insects very often serve as vectors for viruses. So even a very low level of infestation can create a lot of problems because once your plants are infested with a virus, um, there's no cure. Yeah. And so you definitely, as a grower, you definitely should scout for for these insects as well very carefully and take respective measures if you find them on your plants. Because any of these little critters can can really make your plant sick, and then you really got a problem there. Um, Maybe
Uh,
not because of the THC level, but maybe because of the status, overall status of your plants, basically, and your entire facility. That's the other thing.
sure. And in general, pathogens are pretty devastating to crops, but have you seen any research of pathogens increasing secondary metabolite production?
I personally not, I mean, we never worked with pathogens.
Okay.
I'm not necessarily aware of a lot of literature, but I don't want to exclude it at all. Again, terpenes definitely have an effect on pathogens. We're currently working on something similar here in collaboration with another group here with tomatoes. And there's plenty literature um, where terpenes have been shown to have badly, in general, antimicrobial activity, which includes bacteria and fungi as pathogens. So I'm not aware that cannabinoids are having similar effects, but I don't want to completely exclude it. I'm just not aware of it. But the terpenes definitely have quite dramatic effects there. So that could very well have an effect in hemp and cannabis as well.
Now, cannabis seems to respond pretty well to pretty aggressive pruning, mechanical pruning. Uh, would that I'm learning today about how a plant can sense all of these other aspects of herbivory. So I had assumed that when we were pruning heavily, we were mimicking at least certain aspects of herbivory. But what are your thoughts on that? I guess can we create these similar mimicry?
So any type of wounding of of a plant in principle kind of has somewhat of that effect or can have that effect. But one thing you have to keep in mind is that if you do pruning or do by accident wound your plant one time, that might very briefly induce something, but a lot of this is happening on a time scale basically. So if you wound a leaf, you might see a little induction, but that drops back down to baseline level very quickly I wouldn't say within hours, but within a day or so, very quickly. So for a more substantial upregulation, you would have to have a little bit more a persistent and an ongoing sort of process. And that's normally
Hmm.
what you rather face if you have an infestation with a herbivore. Yeah, a herbivore normally doesn't just come by, bite in one leaf, and then move on and go somewhere else. It normally stays on your plant for quite a bit of time. And unfortunately, a lot of insects have the ability to propagate very quickly. And so you're not going to have just one insect nibbling on your plant, but in no matter of time, you're probably going to have multiple insects nibbling on your plant. And then that is this persistent, ongoing herbivory. And that, of course, leads to a completely different response, yeah, from the extent
Yeah.
that
Okay.
you're
That's
seeing.
really interesting because when I think about it, like as a indoor cultivator, we tend to have to do defoliation of most genetic cultivars just to increase the amount of light and air that's getting the plant and maximize our photosynthesis and reduce the risk of pathogens and pest pressure by having microclimates inside our canopy. Um, but what you're saying is when we do that, when we say certain plant responses, but they're short lived, whereas that insect is going to be constantly nibbling. And so it's essentially just keeping the plant stressed the entire time. So you see
Times, yeah.
upregulation of that over a longer period, which is more likely to lead to these
Yeah.
increases in metabolite production. Ah,
Exactly, yeah. So I one-time pruning, yeah, you might see a little bit of an effect there, but it's not going to be very persistent. Versus if you have a pest infestation, which normally lasts for quite a bit of time, then you're probably going to see much more of it. The other question, of course, is also at what time of the sort of the growth cycle are you going to have that? Yeah. I would assume your pruning you do not on the very last stage of before you harvest. You probably would do that sometimes earlier because you kind of like you described, You kind of want to get maximum light exposure. You want to have airflow in there and maybe avoid these microclimates, et cetera, et cetera. You're probably not going to do that in the very 2 days before you harvest. Yeah, you're going to do that while you're cultivating, while you're growing. Um, so as long as you do that sort of in the more vegetative part of the growth stage, you're not going to have much of an effect. Yeah. But if you have that in the very last stage, shortly before you start harvesting anything, then of course, and then if it's extended, then you might see much more of an effect. Yeah. So it really it's the
Yeah,
timing and the frequency of sort of these events happening that might have an influence there.
I think there's a balance there, and we do a pretty big defoliation, say like week 3 of flower, week 2, right around there. But then I like to go through and just pick leaves when I'm in my grow, um, as I'm going through it, because the plants always bounce back so quickly, and I wonder if that might actually have some benefit. Though the downside of this, just to make people aware, when you're doing any sort of wounding, you are opening yourself up to greater risk of pathogens. You've created a perfect place for pathogens in your plant. So there's pros and cons to it, I suppose. We may see increased benefit from herbivory, but we may see, a higher risk of pathogen pressure,
Well,
and
on a
infection.
very practical side, I mean, if you're using tools for doing your pruning, you want to be very careful because you could be spreading diseases from one plant that is infected, to others just using the same clippers. Yeah.
Absolutely. Yeah, that's
even
a very
without
good point.
even without any pests. So I mean, like we're always very careful with these sorts of things. I mean, you want to wash and sanitize your tools that you're using in your growth facility if you're moving around and you're maybe already suspecting that you have something that's diseased somewhere in there. Because otherwise, you don't need any other factor for spreading things. It's actually you spreading things from one plant to the next.
Yes, and insects can be a vector too for folks. So, I do have a couple other questions. I want to totally switch gears, but I'm looking at an article about you in greenhouse management online here. And one of the things that it mentions is that your lab was studying interactions between mycorrhizal fungi and plant roots and how that impacts cannabinoid production or accumulation. Can you just touch on that? Because I know we've been talking for a while. Maybe I'll have to have you back on, but can you
Yeah.
touch briefly on that?
Well, that was one idea initially, and that really didn't get too far.
Mm,
We had another group here in my college here that is working on mycorrhizal fungi. We actually for quite a number of years we were hosting what's called INVAM, which is one of the largest um, even national and international collection of mycorrhizal fungi here. In the meanwhile, since the faculty that was in charge of that retired, that has moved on and is now in Kansas. Um, but we still had some of that collection here, and we were just trying to play around with that to see if mycorrhizal actually how good they interact with hemp or not. And we ran into quite a bit of roadblocks there. I'm not saying it doesn't work at all, but that was kind of not particularly too easy to work it out. In particular, because for the purpose of how we need to do these inoculations for the research side of things, the growth conditions the hemp plants really didn't like particularly. So I'm not saying that mycorrhizal fungi would not interact with the roots of hemp plants, but under the conditions that these folks here wanted to grow the plants for inoculation, the hemp plants just didn't like the condition particularly. So we kind of gave up a little bit at some point following up on that. I can't really tell you much about that. What I can tell you, what we're currently doing, and that follows a little bit more on along the lines of what I was describing before with herbivory. We're currently looking into interaction of nematodes with the roots of hemp plants, because
oh,
that could lead to a very similar response as the herbivory. And we have some promising results in that. We're following up on that right now, and I hope in the next couple of weeks we can actually collect some more samples and then do some more analysis on that. So we're looking a little bit into that because that could be another point where you could have one of these biotic interactions. And I mean, you're probably not going to see nematodes if you're growing in a controlled environment. But if you have your hemp or cannabis plants growing in a field situation, you're definitely going to see nematodes. I mean, there's nothing you can do about. Um,
What
and that,
species of nematodes? Are you talking like root knot nematodes or
yeah,
what?
exactly. And we're testing a couple different ones, but yeah, that's one of them we're
Mm.
testing there. So, and that is a little bit on the backdrops, and I'm going to go back a little bit to what I said before. So once we had seen with the insect herbivory that we had this massive regulation of some of these minor cannabinoids, we kind of started thinking a little bit of how this actually makes sense. And one of the things that we did is that we actually looked into other plant tissues than you would normally do. So most people probably know, I mean, of course, we're all aware that cannabinoids are formed in trichomes of the flowers. There's also trichomes on leaves, and they do produce some cannabinoids. Also, the profile is a little bit different than in the flowers, but there is, and the levels are much lower. But we basically systematically, a little bit more systematically, looked into different plant tissues in hemp plants as to if they would be producing cannabinoids. And long story short, yes, there are cannabinoids in other plant tissues. It's not just flowers and leaves. And knowing that, now it starts making much more sense. So we also found some cannabinoids in roots. Yeah. And that's a little bit the background of that. So now we're that maybe the cannabinoids in the roots also have an effect on
Hmm.
biotic interactions in the roots. So we can't yet confirm that finally, but I mean everything that we have points a little bit in this direction. Um, and so that's why we're following up on that. And in particular, it seems to be not the usual major cannabinoids that play a role in these things, but it's rather some of the minor cannabinoids that play a role there.
Hmm.
So what could be is that some of the major cannabinoids are actually there either due to human cultivation or more for interaction with mammals, and that some of these minor cannabinoids are actually there for interaction with insects and other non-mammalian herbivores, if you want to phrase it that way. And then if you consider that, it would make perfect sense if some of these compounds are being produced in other tissues that, of course, are being exposed to herbivory similar to leaves or flowers. Yeah. So that's a little bit where we are right now. And so,
Well,
yeah,
I
sorry
can't
that,
wait to see what your results are on that. That's really fascinating. Yeah, that's amazing. Not something that was even on my radar in terms of thinking of like root herbivory from the perspective
yeah.
of
Well, I mean, one
nematodes.
other thing that is becoming clear now, and that was kind of one of the big questions on the science side of things, is a little bit um, why are we aware of cannabinoids just in the context of hemp and cannabis plants? And or are they indeed the only plant producing cannabinoids? And that has a little bit to do with the biochemistry of things because the building blocks that you need to make a cannabinoid compound are actually present in principle in any other plant as well. And so it's for us on the biochemistry side, it was a little bit of a puzzle to accept the fact that cannabinoids would be really just produced in cannabis plants and not anywhere else.
Ha-
And actually, in the last couple of years, literally the last 3, 4 years, there have been a few publications now
yeah.
from other plants where cannabinoids are being found. So it seems to be not just cannabis, but other plants as well.
And fungi, correct?
Yes, there are some other organisms that can do it, but in plants there are some other plants, and they're not related to cannabis. That's the other thing that's kind of
Oh,
interesting. So and that's kind of was one of the questions out there that we were also kind of, I mean, we're not working on this currently, but of course we're following this literature and this area quite interested, in particular with in knowing what we found with respect to herbivory, where we're like wondering, well, if that's if you can produce it in other plants as well, then it might become much more interesting. So what we actually, and I think I mentioned that in our last meeting already a little bit, that we have recently submitted a provisional patent on some of these minor cannabinoids due to their insecticidal activity that we discovered. Because we think that if they really have this activity as we've found, this could be something where we could actually use some of those as an organic insecticide, basically. And you could also go down the road and basically not breed, because of course you can't breed if it's not a cannabis plant, but you could genetically engineer it in other plants and basically by that protect other plants through cannabinoid formation.
So you're saying that if cannabinoids truly are a response to herbivory and may offer some sort of insect protection, um, you would expect to find it in other plants too, because other plants would have evolved to have this. And now you're saying that in recent years we've discovered that it does exist in other plants, and so there's just stronger evidence that the possibility these minor cannabinoids may play a role in plant defense, and that down the road we may be able to mani- manipulate that
Utilize
or
it.
understand, utilize it. Thank you. That's the word I was looking for.
Yeah.
Okay. All right. Last question though, because we've already gone over our time. You mentioned this, and I hadn't heard this, that you can use terpenes as a biofuel additive in this article. Can you just, in a minute or two, just touch on that? Because I found that real fascinating. I had never heard this.
Okay. Yeah, that's a completely different avenue. So
Totally.
yeah, there is quite a bit of interest in terpenes as sort of a fuel, either as an additive or even as a pure fuel by itself. The simple logic behind it is terpenes are volatile compound. They're scent compounds. So they from their physical chemical properties go emit into the atmosphere at relatively low temperatures. Yeah. So barely at ambient room temperatures, they're emitted into the gas phase, and that's why plants are using them as a way of communicating, basically. Yeah. But that's a property that you want to have for fuel as well. So one of the problems with biofuels is that, I mean, if you take your canola oil as a biodiesel, if you put that in the tank of your car, and in winter the temperatures are low, your oil starts solidifying. Basically, your vegetable oil turns into margarine, yeah. And of course, you can't drive on that, yeah. That's a problem because it's not liquid anymore. And the same problem you're also facing if you think about what's called sustainable aviation fuels. So when you're flying a plane at 35,000 feet altitude, it's minus 60 degrees out there. And of course, any fuel that you have in your tank of your plane also needs to stay liquid, because otherwise you can't fly in it anymore. And so that's with kerosene, that's the case. And so that's the same thing when you want to have replaced kerosene with something more sustainable. You have to make sure that whatever fuel you're using, it stays liquid at low temperatures. Yeah. And terpenes check mark that box, basically. You can really chill them down to quite low temperatures and they're still liquid. And so either by using them purely or by mixing them in, and by that basically also lowering the solidification, so basically the temperature by which a fuel
Freezing
solidifies.
point.
Yeah, the freezing or melting point. Yeah.
Yeah.
Um, So basically, you can do a mixture or a pure. So there are actually this, and this is actually being done already. So even the U.S. military, the Air Force, has already done and flown planes on terpenes. So they're using it as an aviation fuel already. I mean, of course, this is experimental. This is not a final applied state, but it is definitely an idea that is more than just the brainchild of some weird scientist. So this
Yeah.
is really has some real world applications ready. And it basically the idea is like, I mean, we can drive our cars with electricity now. We can replace fuel consumptions with basically electricity, and hopefully you get that from renewable sources. But you can't fly a plane on electricity, at least not for very far. I mean, short distances you can do, but international aviation doesn't work with electricity. Yeah, that it's just not doable. And so for certain purposes, you still need a fuel, a chemical. And if you want to do that a little bit more sustainable, then you have to come up with ideas. And terpenes are one of those things that's out there, sort of in the portfolio of ideas that are currently being considered and tested out. And I mean, bringing it back to hemp, I mean, they're producing a lot of terpenes. And so it could be in the long run an interesting crop where you could potentially produce terpenes that then you could use as a fuel additive there later on. So this is one of the other ideas or reasons why we're interested in metabolism of hemp and cannabis, because there could be completely different applications than what we're thinking about right now. And that could be one of those. I'm not saying that we're going down that road at some point, but
Yeah. I just hadn't heard that. It's fascinating, and I just wanted to, hear your thoughts on it. So thank you for sharing, and thank you for your time today. I have a better understanding of plant stress and herbivory, and yeah, thank you for the research that you're doing.
sure, yeah. Glad to be on. Thanks for the invite. Hope it helps.
All right, have a wonderful day.
Yeah, you too. Good talking to you. Take care.
That was Dr. Michael Gutensohn, and you were listening to the Cannabis Cultivation and Science podcast. Don't forget to check out the podcast page at www.kisorganics.com. Just click on the Learn tab and then podcast. And a friendly reminder that we do have a Patreon channel with a free 7-day trial and tiers as low as $5 a month. We do book clubs, special cultivation tips and tricks, monthly video hangouts to connect and share your grow, and even give out seed packs and other gifts at certain tier levels. Your support of Patreon or sourcing your products through Kiss Organics is really what supports the podcast, and we really appreciate it. We also have the upcoming Cannabis Cultivation and Science Conference in Bozeman, Montana this year in September, and there's more information and tickets available on our website. Thanks for listening.